Hi, I'm Brendan. My poster outlines our systematic assessment of a simple question. Does gene expression response to a particular ligand change in different cell types? We asked this question because we're interested in predicting how cells talk to each other from single-cell RNA-seq. And currently we can only hypothesize that cells may be interacting based on the expression of cognate ligand-receptor pairs in pairs of cell types. If we knew what changes in gene expression we'd expect to see if a particular ligand had been activated, or a particular receptor had been activated, we'd be able to improve these predictions. In fact, some methods already do this, making the assumption that transcriptional response to signaling is generalizable across cell types. We aim to test that assumption here. We used Connectivity Map, a pharmacogenomics resource that includes gene expression assays of 295 ligands in 9 cell lines. If you average gene expression change across all 9 cell lines, those genes responding consistently will have higher magnitude Z-scores than those responding differently to different cell lines, and that's what we're doing here. And then we feed that into this heat map. Only 1/5 of the ligands assayed had any genes that showed consistent response, that is, these rows of this heat map here. 1 in 20 ligands caused consistent differential gene expression of more genes than we'd expect by chance, and that's shown in red over here. In other words, very few ligands caused consistent changes in gene expression across different cell types. However, if we do the same analysis after averaging Z-scores across samples within each cell type, and so now we have cell types in rows and genes on the columns here, 8 in 20 ligands caused differential gene expression in more genes than we'd expect by chance in at least one cell type, consistent across different dosage and duration of ligand treatment. And finally, this box plot is the culmination of all of this data, and it shows the distribution of p-values when testing against hypotheses that ligands are causing changes in gene expression of more genes than expected by chance when averaging across all cell lines, that's in red here, or within each cell line, that's in green, or specific to each treatment condition, so that's cell line, dosage, and duration of treatment, and that's in blue. Irrespective of the threshold used for defining differential gene expression, the connectivity map data suggests that ligand response is consistent only within each cell type. Since there are limitations to the connectivity map assay, we used a collection of individual microarray experiments to corroborate this finding. Rank correlation of ligand-mediated change in gene expression was significantly higher in the independent assays from the same cell type versus across cell types as seen in the first figure. Overlap of these significantly differentially expressed genes was also higher within the same cell type versus across cell types, robust to the thresholds used to define significance. In conclusion for this part, independent data sets confirm our finding that transcriptional response to ligand stimulus is consistent within, but not across, cell types. However, it's not all doom and gloom for cell-cell interaction prediction. So, different cell types respond differently to the same ligand. Does the reverse hold true? Pairs of cell types exposed to the same ligand were compared, both by correlating their untreated transcriptome on the x-axis and the differences in gene expression upon ligand treatment on the y-axis. The strong correlation between transcriptome similarity and ligand response suggests that similar cell types share their response to ligand stimulus. Therefore, in conclusion, while generalizing ligand response signatures across cell lines is not possible, we may be able to extrapolate from known similar cell types and use these signatures to improve cell-cell interaction prediction.